Good afternoon, everyone. My name is Brooke Garland, and I will be taking you along on your introductory course to Phage Therapy 101. So, as you see, what is phage? Well, bacteriophages, also known as phage, are a type of virus that infects and replicates within a bacterium. And it is also found everywhere. Wherever bacteria is, there are phage found there as well. It can be found in your soil, in plants, animals, humans, and even in our oceans. So they are also composed of a protein, capsid head, as you may see in this picture to your left. It holds the RNA or DNA genome, and it is also found to be in caudovirulase, which is a type of morphotype that they can have, seen in Slipoviridae, Myoviridae, and Codoviridae, which is shown in the diagram above. They also have two life cycles in which they are also able to attack bacteria, known as the lytic cycle and the lysogenic cycle. With lytic phages, bacterial cells are lysed and destroyed after immediate replication of the bacteria. As soon as the cell is destroyed, the phage progeny are able to go to a new host and infect it. In contrast, the lysogenic cycle is not able to immediately lyse the cell. Instead, its viral genome will integrate into the host DNA genome, allowing the virus to reproduce in all of the host cell's offspring. It'll be dormant during the time that it is in there until host conditions change, in which it will then proceed to a reproduction cycle and then lyse the cell just like in the lytic cycle. So, now that we know a little bit about background of what phage is and how it attacks the bacteria, we need to know who discovered it. Well, in 1915, Frederick Tourt, a bacteriologist, actually discovered phage by describing a glassy and transparent transformation called plaques, where it was planted lawn of bacteria that did not grow, that you may be able to see in the photo above right there. And in 1917, Felix Durrell, a French-Canadian microbiologist, discovered phage. And the first known therapeutic use occurred in 1919 when Durrell experimented with phage therapy to cure dysentery in children. As a result to the development of antibiotic resistance since the 1950s, there has been a renewed interest into the ability of phage therapy to kill bacterial infections. So in fall of 2020, I participated in C-Phages, which is a 2-semester discovery-based undergraduate research course that begins with simple digging in the soil to find these viruses, but progresses through a variety of microbiology techniques and then complex genome annotation, which leads to bioinformatic analysis. And this course introduced me to phage and allowed me to make an impact on further exploration of phage discovery and phage therapy advancement. So, as a matter of fact, in this photo here, the phage that you see, that's the phage that I actually found during this course, and you can see it on phagesdb.org, along with other phages from other institutions that students have found. So, why should we care about phage? As the world begins to, why should we care about phage? Okay, so as the world runs out of antibiotics, hospitals will begin to admit patients with infections that used to be easily treated, but now will become fatal due to antibiotic resistance to these infections. And so, therefore, phage therapy will be a very important asset in treating infections in our near future. Some advantages are that they are specific, so species-specific, therefore, we can target specific bacteria with its respective phage. And phage, again, is found everywhere. And C-Phages is actually able to help in finding these species-specific phage. And that could also be a pro and con to using phages. Exponential growth at the site of infection allows smaller dosages to be used, unlike in antibiotics, where you kind of have to take it often, but with these, they have that exponential growth, especially due to that lytic cycle. So smaller dosages are needed, and they can be used alone with antibiotics, or they can be used with other drugs. There we go. So, Dr. Stephanie Strapedy, an infectious disease epidemiologist, and her husband Tom Patterson took a trip to Egypt in 2015, in which her husband became very ill, diagnosed with pancreatitis, and later developed an abdominal abscess in which they found Acinetobacter baumannii. Very long name. It's also known as Arachibacter, but we're just going to continue to call it superbug. As weeks turned into months, Tom began to develop organ failure, multi-organ failure. He fell into a coma, and doctors believed that he actually would not be able to make it. But through Dr. Strapedy's research, she discovered phage therapy and began on her own phage hunt, contacting phage scientists, and that would soon reach to FDA's approval of being able to use phage therapy on her husband, as it wasn't used in the US at the time. And the US Naval Medical Research Assistance in finding a phage that would be able to kill this bacterium that was in her husband. But luckily, Ryland Young, a biochemist at Texas A&M, actually did just that. He found the bacterium that would be able to help kill the bacteria in his abdominal abscess. So a purified cocktail was created from Young's lab and injected into the abscess with the superbug and actually through his bloodstream, which has not been done either. So it was, 3 days later, he actually did wake up. So that was great and a blessing because he really was on his deathbed. But months later, he was able to be released from the hospital, and then he fully rid of the superbug later on. He became the first person to receive intravenous phage therapy to treat systemic superbug in the US. And their story reached so many people and helped other bacterially infected patients seek phage therapy. Dr. Strapedy also shared her story on TED Talk in 2017, which is why TEDx is up there. And they published a memoir named The Perfect Predator. So in conclusion, there is still much to be done in researching bacteriophages. However, with continued study and awareness of phages and phage therapy, we can make further advancements in the future for future therapies against infections where antibiotics will no longer be effective. So that was my talk.